Sir, good afternoon, sir.
Yes, good afternoon. How are you?
Fine, sir. Fine, sir. Sir, welcome to the FDP program, sir.
Thank you very much.
We are going to start the session in a couple of minutes, sir.
Oh, yes, it's all right.
Yes, sir. Thank you, sir.
So shall I start, sir? Shiva, sir?
Yes, madam. You can start now.
Let us start, no?
Yes, yes.
Yes, thank you. Honorable Speaker, Dr. Asadulghani, respected principals, organizing secretary, distinguished members of organizing team, dear participants, a warm welcome to you all for the 6th day of International FDP on recent trends in transforming life sciences, focus on academia and industry symbiosis. The theme for today's session is biosafety and biosecurity. So in the face of current risks and challenges, the international community must work together to avert the threats and to safeguard the global biosecurity. In this context of current situation regarding biosafety and biosecurity, the current session will be a good opportunity for all of us to understand the risks and challenges posed by major infectious diseases. This session will enlighten us with more insights in the field of biosafety and biosecurity. We are very fortunate to have the renowned speaker in the field of biosafety and security, Dr. Asadulghani Garu, with us for today's session. Once again, a warm welcome to you all for this 6th day of FDP program. Have a knowledge enriching session ahead. It's my privilege to introduce Reverend Sister Velanjani Kumari Garu, Principal of St. Paul's Degree and PG College for Women. Madam was a passionate teacher of English. She is a member of many professional bodies. Under her able guidance and dedicated support of the entire staff, the college has obtained A+ grade with a 3.38 CGPA by NAC the year 2018. So I request Madam to address the participants. Thank you, ma'am. Now you can continue, ma'am.
Thank you. Thank you, Pushpalata. And thank you. A very good afternoon to all the participants on this virtual platform. I deem it a privilege to greet all of you and meet all of you on this virtual platform. Thank you for making us part of this International Faculty Development Program on recent trends in transforming life sciences, focus on academia and industry symbiosis. I'd just like to introduce St. Pius X Degree and PG College for Women, which is located in Hyderabad, which is running 19 programs in UG and PG. As Pushpalata have already informed that the college is accredited with A+ grade by NAC in 2018, and we are heading towards autonomy. And we have Biotechnology Department, which is a very vibrant department. And it's our pride to share that the department, the students from life sciences got an opportunity to undergo a unique pilot project training program for 50 hours framed by task, Telangana Academy of Skill and Knowledge, and 4 industries of Hyderabad, namely Shanta Biotech, Bharat Biotech, and Biological E Limited. The students were trained under them, under the pilot project as part of the program by the Biotechnology Department. And I'm sure that the students of St. Pius College and our college is eagerly waiting to collaborate with the reputed industries in future for carrying out research work and placements. Thank you for giving this opportunity, and I wish all the participants a great learning day ahead. And thank you for making us part of this. Thank you. God bless.
Thank you so much, ma'am. Now I request Dr. C.H. Shirisha Madam to introduce the speaker to the audience. Shirisha Madam, it's over to you.
Thank you, ma'am. It's my great pleasure to introduce eminent speaker, Dr. Asadulghani, President, Federation of Asian Biotech Associations, Head Biosafety and DSL-3 Laboratory, Bangladesh. Dr. Asadulghani has received his Master of Science in Biochemistry and Molecular Biology and joined the Molecular Genetics Laboratory of ICDDR-B in September 1995. He received a Master of Philosophy in the field of molecular genetics in 2001. He received Monbu Kagakusho Fellowship for PhD in Japan 2001 and completed his PhD study from Saitama University, Japan in September 2004 in the field of biological and environmental sciences. He received the postdoctoral fellowship under the Japan Society of Promotion of Sciences to work in the field of genomics and bacteriophage biology in pathogenic E. coli strains. After his postdoctoral work, he was offered to join the university as an assistant professor for the Department of Infectious Disease, Division of Microbiology, Miyazaki University, Japan. In July 2019, accepting the offer to work at ICDDR-B again, he relocated back to Bangladesh and initiated the center-wide Biosafety and Biosecurity Program from September 2009. Currently, he is a full-time biosafety professional working to develop and implement policies and procedures of biosafety and biosecurity in laboratories and field activities of ICDDR-B and in the entire country as well. Under his leadership, Bangladesh Biosafety and Biosecurity Society was formed in November 2011. He served for 2 terms, 2016 to 17 and 2018 to 19 as the Executive Committee member of Asia Pacific Biosafety Association. 14th PBA International Conference on Biorisk Management and the pre-conference workshop was organized in Bangladesh under his leadership in 2019. He also served for International Federation for Biosafety Associations as a member of the Board of Directors from 2017 to 2020. He also served as the head of education section of a global network of Bangladeshi biotechnologists. And currently, he is the general secretary of the network, where he is also trying to introduce biosafety and biosecurity in biotechnology for the nation using this greater forum. We will turn the time over to Dr. Asadulghani. Sir, mute. It's in mute, sir.
Oh, thank you. Thank you very much, Dr. Sirisha. Thank you, Kushalata, and also my colleague, Dr. Sambashiva, for inviting me for this talk. Yes, biosafety and biosecurity. I hope everyone can see my slide.
Yes, sir.
Okay, thank you very much. Yes. So this is an area, biosafety, biosecurity, we say this is knowledge-based science, and we have to learn and implement in our work. I will discuss, I will present my today's presentation under different sections: biological risk assessment, biosafety, biosecurity, transforming life sciences industry, outlook in 2021, culture of biosafety, biosecurity, and responsible conduct, and implementation of biorisk management program. This is a common picture of risk assessment. So, and this is very common. And we also know the culture of risk perception, assessment, and management in our part of the world in this Indian subcontinent. We have many limitations. So most of the time, our perception assessment is not that much. We are not always prepared, and our management is always with different laps and gaps. And in the field of biosafety, biosecurity, yes, we have many things to do. Biorisk assessment. If most of the time, we think that biosafety, biosecurity is relating to infectious diseases, but in fact, it is not. It is relating to any biological hazard. But if we think about, focusing on infectious diseases, we are working in the laboratory with one biological specimen. Then what we do? We assess the risk of presence of different microbes, agent 1, agent 2, agent 3. So if we think about the blood, we are dealing with blood, we are conducting fasting blood sugar test, and then I'm dealing with blood. What are the target agents for which we need to assess the risk? What will be the likelihood for that agent, and what will be the consequence? And then we need to assess the risk on different techniques. What technique we are going to apply? Are we going to conduct centrifugation? Am I going to extract nucleic acid? Okay, and again, consequence. We have to think about the consequence of different agents. Okay, so this is actually the biological risk assessment on an average. Why do we need risk assessment? We need risk assessment to reduce and minimize the risk of exposure to workers and the environment. And we have to remember that we cannot make risk zero. Okay, risk will never be zero. What we can do? We are working with minimum risk. We have to minimize the risk. And during our work, every time we have to ask ourselves whether we are minimizing the risk or not. Okay. Yes, biological risk assessment. In the latest version of BMBL, previously we were following a 5-stage risk assessment, and latest version of the BMBL, it was published in the end of 2020. And in that, it is mentioned that 6 stages of risk assessment. Number 1 is to identify the hazard, and number 2, identify the procedure. Identify the hazard. If I'm working with Mycobacterium, the hazard is Mycobacterium tuberculosis, and I may contract TB. Identify the risk, the procedure. Identify the procedure risk. Procedure hazard. I am conducting, I am fixing the bacilli on slide for microscopy. So there is one kind of risk. And using the same biological specimen, I am going for culturing microbes. Risk will be million times simplified. One bacilli, we say that one Mycobacterium tuberculosis is stronger than a bullet. So if I am culturing, I am generating from a few number of cells, I am generating millions of cells. So risk is million times simplified. Then the question is whether I can conduct these two kind of experiments, fixing slides for microscopy and culturing under the same biosafety level or not. So this is the third point of assessment. And then I have to review all my work with a subject matter expert. We are, very much familiar with the institutional biosafety committee. The work should be reviewed and revised if necessary by the institutional biosafety committee. And also as part of the ongoing assessment of the risk management, the proficiency of the staff should also be, verified. Okay. And finally, the management strategies are revised regularly to reassess the risk mitigation and are updated when appropriate. Grossly, if we define biosafety and biosecurity, and this definition, when 2 years back, even the experts were arguing with this definition. Some experts were saying that, oh, biosafety, biosecurity is only relating to the laboratory activities, but it is not. Biosafety to save human, animal, and entire biosphere from unintentional release of biohazard. So here the key term is unintentional release of biohazard, and biosecurity to secure the biohazard from unintentional release, secure the biohazard from intentional release for its deleterious effect to human, animal, and the biosphere. Okay, so simply we explain, we say that biosafety and biosecurity, they are positioned on the other side of the coin. We cannot separate one from another. Yes, when we assess the biosafety risk, then we think about the likelihood of unintentional exposure. The biosafety risk is assessed based on the likelihood of unintentional exposure, and then likelihood of infection based upon the intrinsic properties of the biological agent and consequence of the disease there, and then likelihood of unintentional exposure, likelihood of infection based on the laboratory procedures in place, and in place mitigation measure. So on this, we used to conduct the biosafety risk assessment. Okay. Yes, and we classify, considering the biosafety risk, we classify the risk group under 4 risk groups. These 4 risk groups I will also explain in my later slide. But here we say that these 4 risk groups are relating to biosafety risk. Okay, biological safety risk. Also, these are also these will also be directly relating to biosecurity risk. Also, as I have mentioned, that these are biosafety and biosecurity are on the other side of the coin. But initially, it is very much helpful if we relate these groups with biosafety risk. Okay, and then comes biosecurity risk. Biosecurity risk relating to, as I have mentioned, it is the key point here is intentional exposure. Likelihood of intentional exposure is relating to biosecurity risk, and again, and then likelihood of infection based upon the intrinsic properties of the organism, and likelihood of infection based upon the security measures. In case of biosafety, we say that the likelihood of infection based upon the laboratory procedures and in place mitigation measures, and here the likelihood of infection based upon the based on the security measures according to the biosafety levels in place. We will discuss soon about different biosafety level. With increasing biosafety level, there is also an increase in biosecurity measures side by side with safety measures. Yes, in case of biosecurity, we used to, in case of biosafety considerations, we classify organisms under 4 risk group, but here we categorize organisms under category A, category B, category C, priority pathogens. Okay, so these are the priority pathogens considering the weaponization potential. Category A is most suitably weaponized. They can be easily disseminated, transmitted from person to person. And category B, they are the moderately, they can be disseminated moderately. Okay, and category C, they have the ability. Category C can be engineered for developing a biological weapon. Okay, so this classification is actually relating to biosecurity risk, weaponization of the organism. Okay, here I have shown some examples. I will not read these examples. I am sure Dr. Sambashiva will share the slides with you, and you can take a look into these specific examples. But one thing I would like to mention here is Bacillus anthracis. Okay, so this is the number 1 from its, from literature citing and also from its application, practical application. This is the number 1 organism with having the highest potential for, weaponization. And this is another classification, select agent, okay? And this select agent, this classification is applicable worldwide, but origin is, as biosecurity is originated from, Western world. This is again mostly applicable in Western world, but we also follow this classification, select agent classification. There are human health select agent, USDA select agent, and overlap select agent. We will also find some examples of select agent here, okay? I will not explain these slides. Yes, the most important points that we should remember on our mind from, that I'm going to show you in a few later slides from here. Biosecurity concerns in different areas of biotechnology. So most of the time, researchers are thinking that this is only relating to microbes, but this is not. In the field of agriculture, the concern is reducing risk of alien or transgenic genes, and at the same time, reducing the risk of microbial contamination in food. Okay, we from Bangladesh, we were exporting food in different countries. Okay, we were sending different kinds of food, fish, product. Okay, and interesting thing is, you may know about the betel leaf. We were sending betel leaf to different countries, and from 2010 this has been stopped because one person in Europe was infected after eating betel leaf, and that individual was suffering from typhoid. And since 2010, it was stopped. We are not, earning money sending betel leaf. So there are these kind of incidences are there. In biochemistry, harmful biomolecules relating to living organism is actually the concern. In the field of biotechnology, the origination of harmful biomolecules or biomolecules within or outside living organism is actually the concern. We are frequently doing this. In case of botany, harmful biomolecules or organisms within or relating to plants. In zoology, harmful biomolecules within or relating to animals. In microbiology, this is the most well-known area in the field of biosafety and biosecurity, harmful molecules within the kingdom of microbes. You can see the picture on the left side. So this is the famous bushmeat. Okay, Africans in some African countries, they are preparing bushmeat in this way. This is the pteropass bat, large, big size, full size pteropass bat. They are making this kind, preparing pteropass bat like this, and they are getting infected with Ebola virus. In the field of ecology, imported life form from indigenous imported life forms not indigenous to the region. So these are the problem in the field of ecology. Okay, in Bangladesh we were importing African magur at some point, and you never know what are the foreign floras they are carrying into the country. In genetic engineering, origination, cloning, variation, upregulation of genes expressing harmful biomolecules is a concern. In medical science, organ tissue transplant, gene therapy products, or microbial infection in human and animals. These are the problem. In exobiology, at some point, until you can say 2019, before the COVID-19 attack, okay, COVID-19 attack, exobiology was the biggest concern for the Western world. So they were thinking that they have controlled everything. Only problem may come if something is coming from the outer space, okay, from outside of the Earth, okay. And they were thinking how to manage this. NASA was thinking about that, okay. But when COVID-19, was initiated, okay, then this has changed the shape of the world, okay. It's a kind of retreat for the entire Earth. COVID-19 is now the largest problem. And now we are in the vaccination program. We are in the vaccination era. And this is again the history, biotechnological advancement. For only, fighting against only one disease, we have more than 100 active vaccines right now. And more than around 9 to 10 vaccines are already available in the market, okay? And then we are again facing new questions. How long the immunity will last, okay? And one thing I would like to remind you, please keep on mind, vaccination is a biosafety program. Vaccination is a biosafety program because vaccination, as I have mentioned in the definition at the beginning, biosafety is the control of biohazard, and that is for unintentional release. Vaccination, if I am vaccinated, it will protect me from unintentional release and exposure to COVID-19, SARS coronavirus. At the same time, vaccination is also a biosecurity measure. One vaccinated individual will not be spreading disease if he or she is traveling from different countries to my land. Okay. And you see here the microorganisms and humans. I am again coming back to microorganisms and humans. We say that yes, on my body at a time, I am harboring more than 8 billion of microbes, and the total number is actually the more than the total population on Earth. Okay, but what I want to harbor only the beneficial microbes, but I don't want one single SARS coronavirus 2 that can create a problem. I don't want to harbor one Shigella dysentery type 1. If I am infected with single bacilli, tomorrow morning I may not be able to speak with you. Okay, I will be busy at home. So this is actually the major concern in the field of life science, the microorganisms. But as I have discussed, so there are even larger concerns. Okay, biotechnology, genetic engineering can make something that may have a devastating effect. Okay, but specifically we will discuss from here relating to microbes and also animal field also. Yes, biohazard. Biohazard is originated from biological source. Any biohazard. Biohazards are originated from biological source, and this biohazard is actually the key point for biological safety. This could be infectious, this could be non-infectious. Infectious biohazard, these are bacteria, virus, fungi. Non-infectious, those are toxins. And we say that biosafety is biohazard control. And for biosafety, we have two principles. Number one is biological risk assessment. We have started our discussion with biological risk assessment. And number two is containment. Based on biological risk assessment, we have to apply containment measures. Okay, and biological risk. So if we think about the type of biological risk, number one may be laboratory acquired infection. I am an individual working in the lab. I may be affected with the biological hazard. Okay, that could be a infectious biohazard, bacteria, virus, fungi. I am working in a lab with snake venom. Okay, I can be affected with the snake venom. Okay, and then risk of infection of the field workers when collecting samples. So this is field acquired infection, and from the field or laboratory workers during conducting their job, they may spread the infectious disease. Okay, and that is actually the environmental spread of the infectious material. That is again another type of biological risk. And to manage biological risk originated from microorganisms, we consider the pathogenicity of the microbes, mode of transmission, host range of the organism, availability of preventive measure, availability of treatment. Okay, so these are actually the things, these are actually the properties, these are actually the parameters for microbes we used to consider for actually the managing the risk originated from microorganisms. And based on this, we classify microorganisms under 4 risk groups. Risk group 1, risk group 2, risk group 3, and risk group 4. Risk group 1, in fact, it is no risk. Risk group 2, they are deadly. Definitely they are deadly. They can cause deadly disease, but we have all vaccination medication measures for risk group 2 organisms. Risk group 3, they can cause serious or lethal disease. We may or may not have vaccination medication for that. But risk group 4, these are the organisms we do not have any treatment, medication, vaccination, nothing. Only known information, if one individual is infected with the risk group 4 organism, 50% chance of mortality. Okay, so this is the risk group 4 as well. Risk group 1. We most of the time say that these are non-pathogenic, but this COVID-19 has taught us a lot. Within few months, we became familiar with something, mucormycosis, okay? The black fungus. These are the opportunistic pathogens. Black fungus could be one best example of risk group 1 microorganism, okay? For immunosuppressive individual, this is actually the causing deadly disease only in case of immunosuppressive individual, okay? And there are other examples of risk group 1, the laboratory strain E. coli. Risk group 2, Vibrio cholerae is the best example of risk group 2. We have vaccination, we have medication, okay, we have the oral rehydrated saline. Only frequent intake of oral rehydrated saline will replenish the fluid loss, and that is actually the best management for cholera. Okay. For risk group 3, the best example could be Mycobacterium tuberculosis and also the Bacillus anthracis that I have mentioned. For risk group 4, yes. For risk group 4, the example Ebola virus, Nipah virus. These are the risk group 4 organisms. Okay. In these two cases, Lassa virus. Okay, and there are a few other examples, not many examples for risk group 4 agents. Now I will stop here for a while, maybe 2 minutes, and I will ask audience under which risk group SARS coronavirus 2 will fall. Anyone? Risk group for SARS coronavirus? I think all of us we know. That is risk group of SARS coronavirus.
Risk group 4, sir.
Risk group 4. Okay, anyone else? We have
risk group 2.
Risk group 2. Okay, I have got one of our colleagues said risk group 2, another risk group 4, and then the question is why? Why it is risk group 4? You are considering it as a risk group 4 agent.
Risk group 3.
Risk group 3. Yes. Yes, actually risk group 3 is yes. It is a risk group 3 organism. It is not a risk group 4 organism because the mortality rate is very low. If for risk group 4 organisms, mortality rate should be more than 50%. And it is not a risk group 2 organism because this is severe, okay? And causing, we did not have, until some point, we did not have vaccination. Active medication was not also there. So this is a risk group 3 organism. And this is not a risk group 2 organism, definitely. Similarly, we in Bangladesh, we are now suffering from dengue outbreak. Dengue virus is also a risk group 3 organism. We had in 2018-19, we had chikungunya outbreak. This is another organism under risk group 3. Thank you very much. Okay, so I am now moving to my next slide. Yes, we can see here a few examples of, these are the most popular examples. Lactobacillus under risk group 1, Vibrio cholerae, risk group 2, Mycobacterium, risk group 3, and Ebola virus, risk group 4. And up to here, we discussed about the risk group of microorganisms, and we were discussing about 2 principles of biosafety, risk assessment and containment. Okay, and after conducting this assessment on different microbes, we have identified 4 risk groups. Okay, and also after conducting a kind of gross assessment, we have categorized biological material as infectious and non-infectious. Infectious materials are, some infectious materials are infectious and hazardous and non-hazardous. And biohazardous materials are infectious and non-infectious. Infectious are bacteria, virus, fungi. Non-infectious are toxins. Okay, and you may ask under which risk group or how to deal with blood or urine or stool. it's a kind of unknown. I mean, it's kind of mixed hazard. There may be there may be the presence of different microorganisms under different risk groups. If it is collected from an Ebola outbreak, I mean, the sample even may contain hepatitis B or C virus if it is blood. Okay, so that could be I mean, mixed risk. Yes. So we have conducted risk assessment on microbes, and we have identified 4 risk group of microbes: risk group 1, 2, 3, and 4. From here, we will discuss about containment, the second principle of biosafety. Containment is principles of holding or being capable of holding or including within a fixed limit or area. This is what we do in the lab. Okay, preventing the unintentional release of biological agents through a combination of laboratory practices, containment equipment, and design and the facility. Okay, and containment has 3 elements. The number 1 is my practice. I am a laboratorian. I have to be performing everything perfectly. Okay, I have to be a perfectionist, and that is the number 1 element of containment. And the number 2 is the safety equipment, the biological safety equipment. I am doing everything perfectly, but when I am dealing with live infectious agent or biohazardous material, I have to do that inside a biological safety cabinet. Okay, so that is the second element of containment. And the third element is this biological safety cabinet, we should be inside a dedicated, inside a well-designed laboratory facility with the laboratory grade facility infrastructure and equipment and benches and everything. Okay, we said that the laboratory room should have the safety module. What is the safety module? Safety module is the closable door, okay, hand washing sink, fire extinguisher. Okay, these are the safety modules. So the laboratory should have the safety module. It should be properly designed for conducting particular kind of work or dealing with particular type of risk group 1, 2, 3, 4, particular type of infectious agent. So that is the third element of containment. Yes. And from here, in a few slides, we will discuss about the safety equipment, the biological safety equipment. Biological safety equipments are classified under 3 classes: class 1, class 2, and class 3. And this class 2 is again subdivided A1, A2, B1, B2. Okay, and this biological safety cabinet is doing its safety job. I mean, for the presence of this HEPA filter, high efficiency particulate air filter, and this filter is capturing all different sorts of particles. And this filter is also considering virus, bacteria as particles. Yes, definitely virus, bacteria. These are also particles. And this HEPA filter is capturing smaller than 0.3 and higher than 0.3 size particles at a much higher rate. But 0.3 size particles are being captured at a rate of 99.97%. Okay, and all the bacteria are above 0.3, and most of the viruses are below 0.3. Only one virus, the pox virus, okay, the variola, has the size around 0.3 microbes. But still, we safety professional, we consider that these virus will be, will be carrying some particles, or when it is in liquid, the size will not be this will not allow the virus to pass through 0.3 micron size. So biosafety cabinet, use of HEPA filter in the biosafety cabinet will ensure the required safety for the users. And we can see here the class 1 biosafety cabinet. This is a very simple type of biosafety cabinet. The air will pass through the hair and then pass out from the biosafety cabinet by HEPA filtration. Here we can see the class 2 A1 type biosafety cabinet. The yellow arrows are actually showing the air circulation inside the cabinet. And this cabinet, this is class 1 cabinet, And another type of cabinet is class 2A2 cabinet. Another type is class 2B2 cabinet. These cabinets are hard ducted, exhausted out. And this is another cabinet, class 2 B1 and B2. This is the B2 cabinet. And this is the class 3 cabinet. And class 3 cabinet is also termed as globe box. You can see here the area of globes, and we can use this cabinet. Putting air through these globes and manipulating samples inside this cabinet. So this is the globe box. And here you can see that different ventilation equipments. And one thing, we should not make a mistake using fume hood and laminar flow inside the biomedical research and diagnostic laboratories for dealing infectious agent. Chemical fume hood is for chemicals. Laminar flow is for clean bench, is for clean work, preparing media. Only biosafety cabinets will be conferring biological safety. Here we can see that different kinds of biological safety cabinet, how they are conferring personal and environmental safety in this table. Yes, we have 4 different types of laboratories, BSL 1, 2, 3, and 4 labs. And BSL 1 and BSL 2 are categorized as basic lab, and BSL 3 and BSL 4 are the containment labs. We can see here the all these 4 types of labs side by side. This is BSL 1, this is BSL 2, BSL 3, and BSL 4 laboratories. With increasing biosafety level, there is an increase in the safety. Different biosafety levels are designed with increasing level of protection against increasing level of risk for the workers. Okay, they may face while with different kinds of microorganisms. When we are dealing with animals or animal research animals in vivarium facilities, we also have 4 biosafety levels, and policies, procedures, principles are same as BSL, but as we are dealing with animal, the facility is termed as ABSL. ABSL 1, ABSL 2, ABSL 3, and ABSL 4. And in case of different biosafety level laboratories also, with the anticipated level of risk, we also enhance the personal protective equipment. Okay, just like in case of BSL 3, we need to use, in case of BSL 1 and 2, we follow the basic PPE, gloves, gown, okay, eye protection. And when we are working in a BSL 2 lab, we can follow the same PPE rules. But in addition to that, sometimes in case of BSL 2 facility also, we may deal with pathogens that may enter into my body through respiratory route, like H1N1 influenza. In that case, we need respiratory protection even in case of BSL 2 facility. And that situation we used to term as BSL 3 practice in a BSL 2 lab. Okay. And in case of BSL 3 lab, yes, definitely our protection, personal protection will be, yes, gloves, respiratory protection, N95 mask or PAPR, okay, and coverall. And in case of BSL 4 laboratory, the PPE will be, in one type of BSL 4 lab, the PPE is like BSL 3, that is called actually the cabinet lab. In one type of BSL 3 lab, we use globe box, but in another type of BSL 4 lab, which is called actually the suite lab, in that case, we need to have the positive pressure suit. So with increasing biosafety level, in this way, PPE also enhanced. Yes, from here, in a few slides, we will discuss about biosecurity. Biosecurity, as I have mentioned at the very beginning, it is relating to intentional release, and biosafety is relating to unintentional release. Biosecurity, has become mostly highlighted in the field of microbes after the 9/11 NSAT attack. Okay, you can see here the letter on the right side. So this was sent by the terrorist to a US consulate. You cannot stop us. We have the anthrax. You die now. Are you afraid? Death to America, death to Israel. Allah is great. So this letter was sent, and these are the spot of Bacillus anthracis spore. And this has created a huge turmoil. Okay, a few individuals died after infected with respiratory anthrax, and after that, the biosecurity policy was strengthened in the US. But similar kind of attack was also there earlier around 1980s, but during that time, biosecurity procedures were not strengthened because that was not, that attack was not with this kind of, Bacillus anthracis type pathogen. It was with Salmonella type. From one laboratory, one person took out Salmonella type and they spreaded that Salmonella type before the day of the election to the food chain of the opposition. Okay, intention was to keep them busy at home. That was also a successful attack, but biosecurity policy was not strengthened at that time because it was with Salmonella. And when this example was established, then biosecurity policy was strengthened. US Postal Department was completely shut down, and it was declared that if you are receiving a letter at home, you have to iron this letter for 5 minutes because the temperature of iron is 200 degrees Celsius. That will kill even the spore of Bacillus anthracis. Audience, I cannot see any one of you. So, Dr. Sambashiva, are we conducting this training throughout these 6 days in front of the black screen?
No, sir, because due to the low bandwidth, some of them are stopping their videos.
Okay. But if you have a question, you can appear on the screen and it will be very much pleasant for you to see.
Definitely, sir, we will inform them.
I can see here Dr. Emmanuel, sir. How are you? Emmanuel, sir. My lecture was so boring, I think.
No, sir, no, sir.
No, sir, not at all, sir. It's very much interesting. We are there, sir.
Thank you.
Nearly 127 participants are
Any
there.
questions so far I have discussed?
Sir, I have a question.
Yes, please.
You're telling about BSL 1234, ABSL 1234. Like
Yes.
that, anything is there for when we are using nanoparticles research?
Yes, when we are using nanoparticle research, actually,
Yes, sir.
origin of the nanoparticle should be considered. If it is from an organism like risk group 3, okay, then we have to follow the risk group 3 biosafety level. But on an average, if we consider biosafety level 2 for biohazards in general, okay, if it is just a particle may cause any effect. If it is biohazardous, if the particle is biohazardous, okay.
Yes, sir.
Not. We are not dealing with the whole agent, risk group 3 agent. Then we can follow BSL2 policies.
BSL2 policies. Okay, right. Thank you so much, sir.
You are welcome. Yes, sir. Sir, I cannot hear you. Please unmute yourself, sir.
Okay. Can you hear me now?
Yes.
Yeah, in the labs now, when we use isotopes, radioactive isotopes.
Yes.
And in 2002, I happened to work in a lab in the United States.
Yes.
There they were periodically, once a week, the university itself, Marquette University itself, they have to conduct biosafety checking. And once a month also the government itself, they send people to check whether the radioactivity is there or not, So that type of isotope also we need to protect ourselves. Sometimes carelessly scientists handle those things and then there's a danger to our health. So,
Right. Sir, actually in the field of, safety science, we used to consider for the laboratories. Laboratory safety program should have, multiple components. Like one is, as with the laboratorians, our intention is to deal with biohazard. So the number one is biological safety. And we biological scientists, we cannot work without any chemical. Okay. Then we need the second component of the safety program is chemical safety. Okay. And then in addition to that, if I have a plan to use radioactive material in my research. Suppose in my case, if you look at me, I have used 32P for 7 years in Bangladesh. I was conducting radio labeling of nucleic acid. And again, when I was in Japan for PhD, there again, for primer extension analysis, gene expression, I have used radioisotope for 3 years. And this is another component of the safety program, radiation safety. So biological safety, chemical safety, and radiation safety. These are actually the 3 different, components of the laboratory safety program. Yes, definitely. If someone is using radioisotope, then where I can remember I was wearing a badge here, and that badge, had a chip, and it is counting the exposure limit. Okay, so every month, the one technician from Atomic Energy Commission was coming to me, and they were taking out my badge, and then they were checking, and it was okay. I was never exposed with high-level radiation, and I believe that because I could continue my work. But yes, there are different incidents also in the laboratories regarding radiation. Okay, thank you very much, sir, for comprehension. Yes, and then I am going to complete biosecurity in this slide. So biosecurity, again, as we have discussed, that biosafety has two principles: risk assessment and containment. And in case of containment, we have discussed 3 elements of containment. In case of biosecurity, we have a few elements of biosecurity. Number 1 is physical security. Number 2 is personal reliability, and then material control and accountability, transportation security, information security, and biosecurity program management. So these are the elements of biosecurity. Okay, we need to implement physical security measures like access control. Okay, the security guard, CCTV. These are the physical components, and then personal reliability or personal security. So the reliable person, only the reliable person should be allowed to work in a high containment facility. And who is the reliable person? We need to do different kinds of policies and procedures to be in place for ensuring personal security. Like if one person has link with a terrorist group, that individual should not be allowed to work in a high containment facility. But we cannot mark that individual as a terrorist. Okay, that individual should not be marked as a terrorist, but based on the background check, we have to select that individual A, B, C can work in BSL high containment facility, but individual D, E, they have some history relating to terrorists or some families, so they should not be allowed to work in high containment facility. Okay, so this is kind of it's a kind of policy and procedures we must follow. And history also shows us some examples. Suppose if I give you an example, in Bangladesh, there was a devastating attack. We say hotel artisan attack. So one terrorist, grandson of one terrorist was the mastermind of that attack. Okay, so if we can identify that kind of situation, we should not allow that individual to work in high containment facility, because there is a chance that something in future can occur. Excuse me. Yes, then material control accountability. We have to be accounted for all the materials in my possession. Information security. Excuse me. Information transport security. Transport security, as we have discussed different classes of microbes, considering the transport security, again, microbes are classified as category A and category B. Category A, we have discussed risk group 1, 2, 3, and 4. Grossly, risk group 3 and 4 are category A, and risk group 1 and 2 are category B. if a biological sample contain category risk group 3-4 agent, that biological sample should also be considered as category A. And these categories are applied for international transportation, air transportation, transportation from one district to another district, okay? And category A has a dedicated package. On an average, we say this is a kind of triple package. And category A package should be, produced by a certified facility. The package should be certified. And category B, we should follow the triple package, but following safety security, if I package category A by category B by a triple package, that is also okay. But in general, we used to purchase both the type of package from certified, commercial provider. Yes. From here, we will discuss about trends in life science, trends in transforming the life sciences industry outlook in 2020. Yes. Various life science industry. Any question and concern? Or it is a background noise, maybe?
Yes,
okay.
sir. Please continue.
Okay. So the various life sciences industry branches may include, but not limited to, pharmaceuticals, biotechnology, environmental sciences, biomedicine, okay, nutracellular, neurosciences, cell biology, biophysics, and there may be many others. And all these areas have specific biosafety, biosecurity concern, because they are involved with biological hazards, okay? And yes, personalized medicine through advancements in genetics, collaborative and innovations of various life sciences companies, advance in drug research, okay? The increased volume of life science research. So all these we are experiencing in 21st century. Yes. From the beginning of the 21st century, rapidly developing field of biotechnology has become integrated with many disciplines that has completely changed the mood of research adapted by traditional life sciences, promoting development and maturations of technology. So if we just think about the development of COVID-19 vaccine, I am really impressed. Even one company in Bangladesh, we are still sometimes I cannot find out Bangladesh on the global map, but we have even developed one COVID-19 vaccine, and that is in trial, okay? And biosecurity and biodefense requires systematic engineering involving monitoring and warning, detection, traceability, prevention and control, diagnosis, treatment, emergency measures, and other technical aspects, okay? So the focus of biorisk assessment and management in 21st century. The focus is the risk and challenges of biorisk management are emerging infectious diseases, biological weapon, bioterrorism threats, misuse of biotechnology, alien species invasion, biological accident disclosure, super resistant bacteria. We are in a kind of era of antimicrobial resistance, okay? So this is my consideration. I am also working in Bangladesh. I am leading the AMR. I'm the AMR PI for different funds. And we say that this is the long-lasting pandemic. AMR is the long-lasting pandemic on Earth. Now we are afraid of pan-drug resistance microbes, okay? Not only one single microbe. So different microbes are getting resistance to different antibiotics, and we are even getting the pan-drug resistance microbes. And the solution for these challenges are improve global biosafety and biosecurity, biodefense capability, establish and improve biosafety monitoring network, improve monitoring and early warning system for infectious diseases, improve the identification, prevention, and control of bioterrorist pathogen. We know the list of bioterrorist pathogen, okay? And that's why actually I have shown the classification of bioterrorist pathogen. category A, category B, category C pathogens. Okay. Improve the management of biotechnology application, improve the technological platform for controlling invasion of alien species, increase research into the mechanism underlying the evolution, transmission of drug resistant strains. Okay, so these are the solutions for the risk. And yes, let us see the culture of biosafety, biosecurity, and responsible conduct. What should be the culture? Yes, the culture is an assembly of benefits, attitudes, and patterns of behavior. Okay, personal standards and ethics designed to prevent loss, theft, and misuse. Okay, so this is actually the culture, and the culture elements are management system, behavior of the leadership of the behavior of the leadership and personal, principles for guiding decisions and the behavior, beliefs, opinion, and attitude. So these are the components, these are the elements of the biosecurity culture. Okay, and that has to be stably integrated. This culture should be stably integrated in all the institutions dealing with biological hazards. Okay, doing research and development with biological hazards. Even conducting diagnosis. Okay. And number one, if we think about the management system should have the specific roles and responsibilities, visible safety and security policy, performance measurement, feedback processes, competency-based training, competency verification, compliance check, and implementation. For compliance check, we have annual safety audit, okay, audits and visits, okay, for compliance check. We need to have the behavior of the leadership and the personnel. Okay, the behavior of the leadership and the personnel. Okay, who is the leader? Who is going to lead the biosafety program? Okay, the individual with proper training, knowledge in the field of biosafety, biosecurity should be spontaneously leading the biosafety program if in a particular organization is missing that component. Okay, and in my case, I did the same. I was appointed as a biosafety personnel at ICDRB. I went for extensive training. Coming back, I have declared myself as the biosafety personnel. Okay, I am now the declared biosafety personnel. I communicate with the top management, formed the institutional biosafety committee, and started the biosafety program. Okay, and then I have started the biosafety program for the entire Bangladesh. And this is the way it is. Principles of guiding decisions and behavior. Yes. Leadership, commitment and responsibility. Okay. Professionalism and competence, learning and improvement, maintaining public trust, and code of conduct. Yes. And then belief and opinion. So this is something really, really very important. Everything starts with the belief. And based on the belief, I used to express my opinion. And belief is not visible. Opinion, yes, it is verbally communicated, but still not visible. What is actually visible? Visible is the result. Attitude is understandable. Okay, and this is actually a kind of dire necessity for a biosafety, biosecurity culture. And let us discuss about how to implement the biosafety, biosecurity, biorisk management program. Yes. So biorisk management program. Biorisk management program has the component of engineering component. We need personal protective equipment. We need administrative control. And one major component is standard operating procedure. For every processes, every procedures, every movement, we should have the standard operating procedure. Okay. And in our case, we also organize training for writing SOPs. I hope in near future, we will be offering such training for all of you. Yes, one complete biosafety biosecurity program requires the following component. Number one is institutional biosafety committee. And I think all of us, we are familiar with that. Institutional biosafety committee will be managed by the institutional biosafety officer or biosafety personnel. And he or she will be guided by the biosafety manual. And his number one tool will be the safety audit. He will be auditing, visiting labs and there will be an annual safety audit. And he or she will have the biological waste management program. Okay. Another most important tool of implementation of biosecurity program is the training. There will be training for every component of safety, as we have just discussed about chemical safety, radiation safety, biological safety. All these will be incorporated. Fire safety training should also be there. Okay. And then we will have a complete biosecurity program. For biosecurity, as I have mentioned, biosecurity and biosafety, these are just on the other side of the coin. There is some limitation. In some language, there is no separate term for safety and security. In those cases, it is a bit complicated. They understand safety security together with the same or single word. Okay. And that's the reason new term has evolved, biorisk management. Biorisk management is actually together biosafety and biosecurity. But what we need to know is that for biosecurity, we do not need a second body for managing the program. Biosafety, biosecurity programs are managed together. These are on the other side of the coin. What is the problem with the lab? In a diagnostic lab, we have practice, that is the number one component of the element. We have safety equipment, we have facility safeguard. And these are the areas where we have concern. Sample receipt, sample process, sample storage. Yes, we are storing sample for 1,000 years. In my case, in my biorepository, I have some samples, HIV samples. The funder requested to store that for 100 years. So this is very funny. And in most of the cases, we don't store sample for more than 5 years. But this sample, I don't know why I have to store it for 100 years. But if the situation is like that, maintaining the security for this sample is a big problem. And also the funding, who will maintain? The project has already been, concluded. And who is going to maintain this sample for 100 years? And this is a biosecurity problem. Conduct diagnosis and generating waste. In the morning, I have started with clean agar plates, and end of the day, I have found millions of infectious virus colony on my agar plate. Now this is the problem, managing the waste. Okay, so waste management is one of the largest point of concern for a biosecurity program. In a diagnostic lab, sometimes problems are very simple. And in case of research lab, just think about the situation. I am working with MDR, XDR, TB. I am conducting research with MDR, XDR, TB. So the problem is here more and more concentrated. And elements of containment should be there, and safety security policies and procedures should be in place for implementing biosecurity program in a research lab. We need to have this kind of risk assessment and management scheme. Okay, identify the agent, what are the activities, hazard identification. If based on hazard identification, risk is determined to be managed, okay, determination of risk, decide or not the risk is acceptable. If the risk is acceptable, we should proceed with the work. If the risk is not acceptable, we should stop work. And we have to have the facilities, we have to have the PP, we have to have the training. Okay, we have to identify the area where there is a lack, and filling out the lacks and gaps, we can conduct that work. Suppose I am now doing diagnosis with SARS coronavirus 2. Okay, and I'm only conducting diagnosis of SARS coronavirus 2. This is molecular diagnosis. We say culture-free disease diagnosis. Okay, and if my facility is for culture-free disease diagnosis, if my facility is for culture-free disease diagnosis, I cannot do culture-based disease diagnosis. And for culture-based disease diagnosis, with a risk group 3 agent, I need BSL-3 facility. If I do not have a BSL-3 facility, I have to stop work at that point. Yes, with increasing biosafety level, what happens? Productivity decreases and operation cost increases. So we should not be thinking that I am working with a risk group 2 agent and risk group 3 will confer me better safety. It will make me a loser. For risk group 3 agent culturing, I will only be going for BSL-3 facility. Okay, even diagnosis of Mycobacterium tuberculosis. If it is culture-free, we can conduct in a BSL-2 facility with BSL-3 practice. Yes, with increasing biosafety level, acceptable level of risk decreases, protection against danger increases. In conclusion, biosafety and biosecurity together will lead to global public health security. And that is actually the most wanted situation, most wanted, policies, procedures for the entire earth. And that is the most wanted thing for the biological scientists right now in this COVID-19 pandemic situation. This is the time. I think I have not exceeded. Am I exceeding the time limit, Dr. Sambashiva?
No, sir. No, sir. No, sir.
Okay, okay. Thank you very much. I am now ready for the interactive session. And all these, we have developed all these training modules with the help of different scholars. Professor Hubert Hens is the professor of Erasmus University. Dr. Barbara Johnson was my mentor. She supported the entire biosecurity program in Bangladesh. She is one of the founder member of American Biosafety Association. And from the very beginning, she is the editor of Applied Biosafety, which is the main journal of biosecurity. And Biosecurity Engagement Program and CDC also substantially supported. These lectures we started to develop, and this was in fact developed support from taking support from BEP and CDC. Thank you very much all.
Thank you, sir.
Thank you, sir. Thank you so much, sir, for such wonderful and informative session on biosafety and biosecurity. So now the platform is open for the audience to interact with the resource person.
Door is open, no one is interacting.
I request the participants to don't lose the opportunity to interact with the resource person.
Good afternoon.
Yes, sir. Good afternoon, sir.
Okay.
Yes, ma'am, you can proceed. Sorry.
Yeah, it's okay, ma'am. Carry on. I'll ask again. Anyway, my doubt till it is clarified, I will not. Okay. Please go ahead.
Okay. All right. Thank you, ma'am. Sir, once again, sorry to disturb you.
Yes, please.
Yes,
Please go
yes,
ahead.
sir. Sir, are you providing any diploma or certificate course on this biosafety for other country? Like you said, you are giving for Bangladesh. So anything is there, like we can get through online, any certificate course or diploma course?
Online course, certificate
Yes,
course.
sir.
Yes.
Yes, sir. Yes, sir.
Yes, we are under FABA, Federation of Asian Biotech Associations.
Yes, sir. Yes,
We are
sir.
going to organize a certification course starting from October 22, and it will continue until 6 November. So I think Dr. Sambashiva will rightly inform you very shortly. And that will be a very good course, I think. We will be covering biosafety, biosecurity in human health, animal health, biosafety in pharmaceuticals industry, biosecurity in clinical care. Okay. And we will also have applied lectures, viral vectors in biotechnology. And many scholars will be coming to present their talk during that certification course.
Okay, sir. So, yeah, sir. It was a wonderful session indeed, sir. Very good, informative session. Thank you so much, sir.
Sir, good afternoon, sir.
Good afternoon. Please go ahead.
This is Jayasuri Kumari from Women's College, Koti. I'm a faculty of food science in Women's College, sir. I'm curious to know what kind of research is being taken up by your organization and country as such in the area of food science at UG and PG level, because biosafety and biosecurity is required for all life sciences, even more for the food also, which we have seen gaining the importance during the COVID period in the last 1.5+ years. So what is the research taken up in that area, sir?
Sorry, what is the research
Taken up in connection to nutrition and food sciences, connecting that to biosafety and biosecurity?
No, in fact, focusing on biosafety, biosecurity particularly, we are not doing any research directly in the field of biosafety, biosecurity. But what we are doing is relating to, biological hazard. We are trying to implement biosafety, biosecurity measures in that, okay? And yes, some research you can say we are conducting some kind of analysis, what biosafety, biosecurity measures throughout the nation we are taking. We are conducting baseline assessment of laboratories, okay? We have around 10,000 hospitals in Bangladesh with medical microbiology laboratories, and we are conducting baseline assessment of those laboratories to strengthen their biosafety, biosecurity status, okay? So this kind of research, we are doing. This is also a database research, and we are also, helping development of those laboratories under different projects, strengthening their biosafety, biosecurity status, okay? And we are, this kind of work we are doing. But specifically, yes, there are, in Western world, they are doing some research, biosecurity, they have biosecurity research center, okay? But we are not, proceeding in that way so far, but definitely we have an interest to do that.
And how about in the food processing sector, sir?
Food processing?
Yeah.
Yes. In food processing sector, yes, we are again, we have some improvement because the, scientists working in food safety, they are also involved with our forum. We are trying to enhance that sector also, food processing sector. Suppose we have the facility that is involved in packaging food before sending to different countries. We are trying to develop their laboratories, okay? In some cases, in the ancient period, it was just a kind of surface checking of the package, but not, the checking inside, what is there. Suppose, as I mentioned, eating beetle leaf sent from Bangladesh. One lady was infected with Salmonella typhi in UK. And after that, we are losing that money. And Bangladesh was earning 10 million, more than 10 to 15 million US dollar every year from that. But we are losing that. So this kind of situation is there. Suppose we can even, we have, you know that our land is so fertile. Just like bacteria, grow so easily. Food, plant, if you just put something on the soil, the plant will grow. Okay, even putting any fertilizer. So our land is that fertile, but we cannot send so many things because of that kind of, issues. So these areas we are trying to, make progress gradually in future. There are some improvement, but it will be visible in near future.
Thank you, sir. Thank you very much.
Good afternoon, sir.
Yes.
My name is Nishat Khatun. I'm a PG teaching faculty. Sir, maybe it's not very much related to your topic, but still I want to ask this question. As developing countries have more threat related to the superbug, are we ready, sir, for any kind of the resistant bacteria, such as E. coli? In what way we can go through with that? Because if you go back to the history before this development of antibiotic or discovery of antibiotics, more deaths used to be because of the infectious diseases. And because of the resistance of these particular bacteria, we are facing those challenges very near, it's in near future. So can you please show me some, strategies which we are going through with that?
Yes. You know that in the field of AMR, antimicrobial resistance, okay, there are some terms: antimicrobial containment, antimicrobial use, okay, and antimicrobial utilization. AMR, okay, AMC, we say antimicrobial containment. So we only think about containment in one direction, but we are not thinking about containment in the laboratory. Okay, in labs also we are doing research. Okay, we are doing culturing different MDR, XDR, PENDR strains. Okay.
Okay.
And indiscriminately we are growing those strains, and are we conducting, we are following the waste management properly to kill those bacteria before releasing the waste? Okay. As a
Yes.
laboratory personnel, we can think in that way also. Okay. And
Yes.
yes, on an average, there should be a kind of overall policies and procedures to manage this kind of risk. Okay, from the hospital to the lab. Okay. And as I have already mentioned, this is the long-lasting pandemic, AMR. AMR is there in E. coli, AMR is there in Salmonella,
Yes.
Shigella, okay, in Vibrio cholerae. Okay, AMR is there in Mycobacterium tuberculosis.
Yes, yes,
Okay.
sir.
So Pandiar is there, is coming out right now. I mean, Pandiar means resistant to all available antibiotics. Okay.
Okay.
So we have to have a kind of integrated plan for management, not only in the hospital. We are just thinking about hospital infection control. That's a smaller area of biosafety. But if we think about the larger, if we think about covering the entire area, then in that case, the term biosafety in clinical care is important. Okay, so this is not a popular term, biosafety in clinical care. Only we think about hospital infection control. If we
Okay.
think about the biosafety in clinical care, that entire thing will be covered under that, larger area. Okay?
Okay. Sir. Thank you so much.
Yes, you are welcome.
So there is a question in the chat box, sir. Can I ask, sir?
Yes, please go ahead.
Sir, it is Dr. Nasrat K. Sheikh. They are asking, can we go for patent application related to bioanalytical techniques, which was developed and validated as per ICH? It's proved safe and effective to suffering from diabetes. So this is the question from Dr. Nasrat K. Sheikh.
Yes. Could you please repeat your question?
Yes. Can we go for patent application related to bioanalytical techniques, which was developed and validated as per ICH? It's proved safe and effective those who are suffering from diabetes.
Yes. Can we go for patent application related to bioanalytical techniques?
Yes.
Which was developed and validated for,
yes.
As per IPC. I am not so much, I think I am not in a position to make any comment.
Can I?
That direction. Yes, I think. Rest, please go ahead.
You need
welcome,
to
sir.
get the.
Suresh, sir, welcome, sir.
Yeah, good afternoon. Wonderful lecture, sir. Congratulations. Actually, it's a wonderful lecture. This particular question actually refers to the biosafety testing analytical procedures. Yes, it can be patented, but it has to get the approval of the CDC and a similar kind of organizations, this one. And it has to be test proven one. That's the answer. I think you got it. It requires to be tested and proved and certified by an authorized team or organization like CDC, Atlanta.
Thank you, sir. I hope the participant is clear with the doubt.
Sir, I have 2 more questions. Can I go first?
Yes, please.
Yeah, Dr. Nasrat, would you like to speak? Yeah, fine. Sir, you said that the black fungus comes under risk group category 1, but in recent developments, what we observed is the patients who are affected with COVID-19, when they affected with the black fungus, the risk factor is more because COVID-19 is comes under risk factor 3, risk group 3. So how can we say that the black fungus comes under risk group 1?
Sir, you are muted.
Sir, you are muted, sir. Please.
Sorry. Yes, so black fungus is considered as an opportunistic pathogen. Okay. So this kind of opportunistic pathogen, those are not creating any disease for healthy individuals, are categorized under risk group 1. So this is number 1 point. So this could be an opportunistic pathogen. This is not creating any disease in healthy individuals. Okay. And in fact, you know that the association of black fungus with COVID-19 is, I will say, it's a kind of misassociation.
Okay.
Because when we are treating an individual with high level of steroid during the COVID-19 situation, COVID-19 infection, the individual became immunosuppressive, and then this opportunistic pathogen is causing, mucormycosis in that individual. And this is the situation similar to high diabetes. In Bangladesh, I was also looking for the information how many individuals are infected with black fungus or mucormycosis. And I have seen that, yes, on an average in a year, 2-3 reports are there, and many maybe unreported cases, but 2-3 reports we have seen only relating to diabetes. Okay. So the association of black fungus is, we are associating black fungus with COVID-19, but I will say this is a kind of misassociation.
Okay.
This is for, indiscriminate use of steroid.
Yes, sir.
Okay. And then how can we protect that? You know that I have mentioned one term, biosafety in clinical care. In that case, we discuss about how one facility should be constructed for treating immunosuppressive patients. Okay, HIV infected individual, we need to treat them in a kind of positive pressure room with HEPA filtered air. But COVID-19 situation is so devastating. Okay, so many people are being infected at a time, and some of them are immunosuppressive. Some of them we are going for treating. We are subjecting them to steroid therapy, and that's the reason. As the number is very high, we are a kind of at a loss. We cannot accommodate everyone in a kind of positive pressure facility, HEPA filtered positive pressure facility. But ideally, if you treat someone, with this kind of medication, they should be accommodated in that kind of room, fungus-free room. If the room is fungus-free, then from where black fungus will infect that individual? So that needs, policies, procedures. We need to revise. We have to be prepared for the next pandemic for this kind of situation. Our medical system, health system should be reviewed. If you go for treatment of an individual high diabetic, or if you subject them to steroid therapy, you have to have that kind of facility. All of a sudden, you can develop a facility in the field with positive pressure HEPA filtered. Okay, so that is possible, but we did not do that. So that's why we are facing. Another thing is that black fungus is the disease of our world, Indian world. Okay.
Okay.
Subcontinent, because another point we have to consider, humidity. It is not the problem in the Western world. Okay. At the very beginning, we did not face it, and when the environment become high humid, okay, then this has happened. So there are multiple parameters. So we have to review our policies, procedures, treatment strategy. Okay.
Yes, sir. Excellent points. So I would like to ask you one more question. So what are the career opportunities in this field of biosafety and biosecurity for the postgraduates and graduate students in the field of life sciences? Career opportunities.
Sorry?
Career opportunities for the students who are pursuing the masters or as well as the graduation in the field of life sciences. If they have done any diploma course in the biosafety and biosecurity, what is the career opportunities?
Yes. Actually, you know that in Australia, USA, UK, there are biosecurity research centers. They are also offering degrees. But in our world, we have not created that. I think I will say we are in a position we can initiate that in India. Okay,
definitely, sir.
Huge area, biosafety, biosecurity. Okay, it's a huge area. It's an applied area of biotechnology. If we overall consider, it's an applied area of biotechnology. So we can create that.
Sir, I'm having one question, sir.
Please.
Water purifiers, nowadays they are keeping the filters and as well as the antibiotics, some amount of that one. Daily ways, when people are drinking that, is it safe for the health of a person, individual? Now, what are the parameters to be considered for the company people who are making that?
Oh, it's very, tough question for me. Yeah, yes. In case you know that you are assessing some kind of threat in water, there may be residual antibiotic or something like that.
Yes, sir.
Okay.
Maybe they are taking that antibiotic doses. Maybe we are prone to maybe susceptible to the particular thing, or maybe the variants we can able to produce, or something will happen. Daily, why to take that much of concentration? What exactly the limit of that? What are the filters? How to control?
And before that, I will ask: are you, drinking groundwater? Surface water or groundwater? Because most of the time I see that we are collecting water from deep from the ground. We are not drinking surface water. So if it is surface water, water from the pond or river, then definitely there is a chance of residual antibiotic and high level of resistant organism. And another point is in the poor world like Bangladesh, we have water supply sometimes contaminated with sewer water. Okay, in that case, we may have this kind of problem. Yes, in some cases, I have also seen the outbreak. Some infectious, some diarrheal disease outbreak from, water is contaminating, our pipeline water is being contaminated with sewer water.
Yes.
So in that case, the problem is there. And I am not, I do not have that much knowledge, in fact, to answer your question, what should be the parameter for filtration. But definitely we can find out the information and share with you later on.
Thank you,
Dr. Sambashiva, please help me to.
Definitely, sir. Definitely.
But yes, definitely we have to be aware that we are not drinking surface water, water from the river or pond.
Yeah.
So that will be, there will be high concentration of organisms and marks and other large particles. But yes, if we are drinking groundwater and if it is, properly pipelined and reaching to my house, it is safe. But still, we consider filtration at different stages.
Because in the valleys, we will consider that water is a pure amount in valleys, and then high altitude, that area, the water, whatever the springs, we will consider that is a pure one. When we are taking the household, the pipes through that, it is coming a lot of contamination. Sometimes floods and other such natural calamities are coming. So again, there is a danger of more.
Right, yes.
Thank you, sir.
You are welcome.
You mentioned about one question I would like to ask Dr. Asadulghani.
Yes, please, sir. Please go ahead.
This is regarding the sample storage for 100 years. I was just thinking, what situation, we have to keep the samples for 100 years, like whether it is a positive sample or the one.
Yes. It was a study on HIV, okay? And at that time, you know that we are storing in my biorepository, that is the only one biorepository in Bangladesh. We have one freezer room, and at the same time, I have two rooms. One room for storing stool and other samples. We are considering those are a little bit below risk, and higher risk samples from outbreak. And altogether, in my biorepository, we have 100 -80 freezer, and most of our 500 liter. And we are storing more than maybe 2 million of samples. And all of those are, our research, we have the RRC, Research Review Committee. They are approving storing samples only for 5 years. And during the time of the project. And everyone has to pay money for the maintenance of the facility. But the biggest concern is with that, because the project was completed and we are storing the sample for 100 years. We are committed to store that. And it's a big problem. Every time we discuss about that, but
it's very difficult one actually.
Yes,
you know that we are building one of the biggest ABSL facility in the country. So when you said that one, it was shocking because but the samples, even if they're positive, and in case they will have to be cryo freeze, or how do you have to minus 190 or so?
Sorry?
No, in case if they're positive, let us say the samples will have to be stored for compulsorily for 100 years. And is it for verification by anybody, or what type of situation will prevail like?
Yes, it is. I mean, that samples are considered as a
only for GLP and all that, we still keep only for 10 years maximum. Like
right. Yes. So that was a strategic mistake. Definitely, I am mentioning every time, because those samples we are maintaining in a freezer room, -20 room. Maintenance of a -20 room, again, it's very difficult.
Yes.
Okay. So continuously, I need to maintain the compressor of that room. So this is actually kind of worst example I show, and it's the biosecurity risk. And every day we need to go there. The security is standing in front of the room. Every hour he's checking the temperature, but it's the kind of commitment with the donor. Okay, that was a HIV study.
HIV. Okay, thank you.
So, is there any more question?
Madam.
Sir.
If there are no questions, I would like to ask you one more question, sir, regarding the biodisposal. I mean, the material that is handled in the laboratory. While sending it out, especially the BSL-3 and BSL-3 kind of facilities, we are building right now BSL-3 facility, which is also in between BSL-3 and BSL-4 kind of facility. We are planning to purchase a kind of kill tanks, which is the mandatory one. Is it a mandatory to purchase the kill tanks to decontaminate the organisms? Because somebody was telling that with holding tank itself, it can be, withstand, I mean, it can fulfill the requirements of the facilities.
So I could not, hear clearly. What tank you mentioned?
Kill tanks, the decontamination kill tanks these days.
It's called a dunk tank. Dunk tank. Is it dunk tank?
Dunk tank is different. Actually, it's a kill tank. The decontamination, the organisms, when you are sending it out, before they actually go into the holding tanks, the material will be processed in a decontamination tank. It's called as kill tanks, actually. KILL only.
Okay.
So this has been recently introduced as a mandatory thing for the people who are handling the BSL-3 and BSL-4 type of organisms.
Oh, okay. I am not aware of that, but yes, ideally, what we implement is that before taking out anything from this kind of BSL-3, BSL-4 facilities, it has to be sterilized and then sent for incineration. So any you follow should be up to the mark of sterilization, and sterilization should be validated. Okay. That much I can say.
Yeah. Okay. This, if it is a liquid material, it has to be sent through this. If it is a solid material, as you said, rightly double bagged, and it will be sent through incineration.
Yes. Yeah. You have to have the effluent treatment plan for that, and the liquid material should pass through the effluent treatment plan. And
before going into the effluent treatment plant, it will be held for some time in a holding tank these days. But recently, even before going to the holding tank, there's one more barrier they introduced. There is a kill
tank. Yes. I got your point. Yes, that is a kind of must for BSL-3 and 4 facilities. Yes.
Thank you.
Sir, what are the consequences of this violating biosafety and biosecurity guidelines, sir?
You can start another pandemic.
That's true, sir.
How we can ensure the safety disposal and follow-up of biosafety security guidelines at different institutions?
Yes, definitely. In different institutions, guidelines should not vary. We should follow the same guideline for all. Okay, and that will bring all of us to a kind of national standard. We should have up to the mark national standard, and we all should be following the same standard. Okay, and that is something important for parallel development and sustainable development. If I say that I am following this standard and he is following that standard, that may create some kind of deviation. So that's the reason we have to follow the same standard throughout the nation. We should have a national standard, and national standard should be complying with. We say that national standard should be, I mean, above that standard. We will do better than them, something like that. That should be the policy and procedure.
Thank you, sir.
Sir, one more question, if you don't mind.
Yes, please.
Sir, nowadays CDs, DVDs, so people are using very badly every area, and they are without having any proper things, they are discarding the material. And they are the centers for the trapping of the microbes. So is there any relevance? This is CDs, DVDs, DVD and CD, whatever we are using. So people everywhere nowadays, common persons also using that. And after certain time limit, they will throw somewhere else. Because a lot of radiation will be there, no, sir. When we are working on that one, we are trapping the energy, we are keeping that. So without proper rules and regulations, we are discarding that. Wherever they want, they are throwing. Maybe they are the centers for the new bacteria and virus and fungus. Can you enlighten us related to that?
Yes, it's really a concern. But if we say that if we follow the hand hygiene protocol, if we keep our hand, because those will be contaminated. If you are using CD, DVD in BSL-3 lab, so that's something different. But if you are using at home, those will be contaminated from contact. So we have to follow hand hygiene, hand washing, keep our hand clean. So that may, I mean, keep everything safe from you. I am the source of infection for that. So I have to keep myself clean.
No, but laymen, sir, auto persons or maybe the bus. So laymen, sir, they are not even understanding that. They are making use of that. Simply they are throwing out once they use that.
In that case, also vice versa. I should keep myself clean. If I touch any CD, DVD, then I will wash my hand and or desanitize my hand before touching my face or mouth or nose. Okay.
Okay.
Excuse me, sir. Might be you have heard about that. It's me, Nishat Khatun again. Sir, did you know that this year Olympic medals is being prepared from e-waste? Japan has used all its e-waste to prepare these Olympic medals. So I think we
from the microchip, yes.
I think that can initiatives we can take to control all these things.
Yes, yes. Reduce, reuse, and recycle. 3 R's.
Yes, yes, sir. Thank you, sir.
Thank you all. Thank you for your wonderful participation. Thank you so much, sir.
Thank you very
much. So now we will conclude this technical session here, and we will have a biological break for 5 minutes, and that will be followed by valedictory session. So I request all the participants to rejoin after the 5 minutes. Thank you all.
Okay, thank you.
Thank you, sir. Yeah.
Good afternoon, Dr. Shiva. Are you there? Can I just take a couple of minutes and just exit the meeting, please?
Just to wait for 2 minutes, sir. We will start the session now. Just 2 minutes, sir. Just 2 minutes.
Yeah. Okay. Carry on.
Yeah.
I'll be just
Yes.
listening. Okay. Go ahead.
Sure, sir. Thank you. Thank you, sir. Thank you. Sir, madam.
Sir,
Start the session, madam. Sir is having some important meeting, so he would like to address. Sirisha, madam.
1 second, sir. I request Shirisha, madam.
Yes, sir.
Yes, madam. You can start the session.
Dr.
Madam.
Sir.
Good afternoon. You can start, madam.
In the end, it's not about the work and not an award you get for the work. A very good evening to one and all, esteemed fathers, speakers, and our dear lively audience. I am pleased to extend a cordial welcome to all of you present here for the valedictory session of the Faculty Development Program titled Recent Trends in Transforming Life Sciences, giving a special focus on academia and industry symbiosis. Without waiting any longer, I would like to invite the Dean of Academics, PG, and FDP course coordinator, Dr. C.H. Sirisha, for the welcome speech.
Sir, ready, sir?
Yes, yes.
Continue the things or shall I?
You start, madam. You start.
Okay, sir. A very good afternoon to ladies and gentlemen. We have come to the end of the event. Time has come for all of us to depart. A warm welcome to galaxy of intellectuals, invited guests, accomplished faculty members, and participants from different prestigious colleges across the globe. Pandemic has transformed the lives of all. We witnessed paradigm shift in all areas of life, embarking a new chapter of life. I'm very much elated to appreciate all the leaders of science who generously shared their valuable knowledge with all of us. The seminar has witnessed insightful comments, compelling arguments, and impactful presentations. I think we all agree that this day is definitely not ordinary. Today, we acknowledge the people met, the lessons learned, the laughter relished, and the knowledge gained. So we savored every event. There is a long way to go. I wish you all to touch a significant milestone on your continued trajectory of success. Thank you once again.
Thank you, ma'am. I now request the chairman of FDP, OSD to VC, Osmania University, Hyderabad, to speak a few words about the FDP sessions.
Please, sir. Please, sir. Access.
Yeah. Sure. Good afternoon. I wish all the participants across the world must have taken the utmost benefit of this 1-week orientation program, faculty development program, on trends of life sciences, the special focus on the COVID pandemics. In a capacity of chairman of this committee, I congratulate the organizers, and I also wish that the entire program could have been gone wonderfully well, and the take-home message from you must have been reaching to the target group. I just congratulate you all for taking part in this wonderful program, and wish you good luck. And I wish that this kind of programs will be happening very frequently in coordination with the different colleges, particularly Loyola Academy and the Government City College and Nizam College. Similar kind of activities will be continuing in future. At this point of time, I only beg apologies for not really spending sufficient time with you because of preoccupied programs. I only can assure and congratulate the organizers for making this program a very meaningful one. Please take care of yourselves and this COVID pandemics, and also convey the message, whatever that it has been learned. And I wish that you will pass on the message to the next generation of your students. Thank you very much.
Thank you, sir. Thank you, sir.
Thank you, sir.
Thank you for the wishes, sir. May I now request our Honorable Chief Guest, Professor P. Lakshmi Narayana, Registrar, Osmania University, Hyderabad, to address the gathering.
Sorry to say, madam, sir will be joining in a couple of minutes. Just proceed with the Dr. Asadulghani, sir. Words.
Okay. So we now proceed with the